You think about those incredible dinosaur skeletons in museums, right? Or maybe a perfect little trilobite fossil. We get this amazing window into the past. But what about everything else, all the other life? Why do some creatures get, like, immortalized in rock and others just seem to vanish completely? What actually decides that?
That is pretty much the million-dollar question, isn't it? And it's exactly what we're diving into today. There's this really fascinating new study out. It's from the University of Lausanne, published in Nature Communications.
Okay.
And our mission, really, for this deep dive is to try and understand the, let's say, hidden rules of fossilization. We want to figure out what makes one organism more likely to stick around than another. It helps make sense of the fossils we do find, why the record looks the way it does.
Right.
We'll be digging into the study itself and also some comments from the lead researcher.
Because you see the hard parts, the bones, the shells.
Yeah.
But yeah, what about all the soft tissue, like muscles, guts? I mean, fossil brains are incredibly rare finds. It really makes you wonder why some things leave such detailed records while most life just poof, gone. And this new research, it seems to point towards something quite fundamental, like the animal's own body.
Exactly that. The core idea they're exploring is that an organism's own biology, specifically its size, and also how much protein is in its tissues, that plays a huge role in whether it fossilizes or not.
Wow, okay.
And the Swiss team, they didn't just, theorize about this. They actually set up some pretty clever experiments to test it.
Experiments. Okay, I need to hear about this. What did they actually do in the lab?
Well, they basically recreated the whole decay process very carefully. They used different modern animals, things like shrimp, snails, starfish, even flatworms. But the really neat part was how they watched them decay. They used these tiny, tiny microsensors.
Microsensors.
Yeah, to track the chemical changes happening right around the decaying bodies. They were especially focused on oxygen levels. Like, is there oxygen present or does it disappear?
So they're watching decomposition, but like chemically in real time. That's pretty cool.
It really is.
And what did they find? That link between size, protein, and the local environment?
The key thing was this. Bigger animals, and the ones with more protein, they tend to very quickly use up the oxygen right around them as they decompose. They create these little pockets of what scientists call oxygen poor, or reducing conditions.
Reducing conditions, okay.
And that rapid drop in oxygen, it's crucial.
Okay, here's where it gets really interesting for me. Why? Why is having no oxygen so important for making a fossil?
Well, think about what usually happens when something dies. Bacteria, other critters, they break it down fast, right?
Right. Decomposition.
Exactly. But if you take away the oxygen, those processes slow way, way down. It gives the tissues a chance. Instead of just rotting away completely, there's a better opportunity for other things to happen, like mineralization, where minerals replace the organic stuff.
So the original material gets swapped out for rock, basically.
Kind of, yeah. Or sometimes the tissues themselves get sort of stabilized or replaced by more durable compounds. Low oxygen helps that whole process.
Huh. So a bigger, maybe more protein-packed creature, it almost creates its own little protective bubble as it decays. Slows things down.
That's a great way to put it, yes. And the researchers, Nora Cortesi and Farid Saleh, they make this point really clearly. You could have two different animals buried right next to each other, same time, same sediment. But because one is bigger or has different body chemistry, it might create those low oxygen conditions and have a decent shot at becoming a fossil, while the other one just disappears. They specifically mention, for instance, that larger arthropods would probably preserve much better than something small and maybe less protein dense, like a planarian worm, just because of this effect.
So the juiciness of the creature itself, its size and protein, is a huge factor. Maybe even more than exactly where it ended up.
It seems to be a really significant internal factor, yes. Which then connects to something else they brought up that I found really thought-provoking. You know how we find so many arthropod fossils from way back, Cambrian, Ordovician periods, like 500 million years ago?
Oh yeah, trilobites everywhere. Arthropods definitely dominate parts of the early record.
Exactly. And Nora Cortesi suggests this might not just be because there were tons of them living back then. It could also be because arthropods, being generally larger and probably quite protein-rich compared to some other early life.
They just had the right biological toolkit to get fossilized more easily.
That's the idea. Their body composition might have naturally favored preservation because of this oxygen depletion effect.
Man, that really makes you look at the fossil record differently. It's not just a perfect census of who lived, but who had the right stuff to become a fossil. Okay, but then, how do you tell? How do we know if an animal, say, a type of ancient worm, wasn't around in a particular place versus it was there, but just never fossilized? That seems like a huge challenge.
It's a critical question in paleontology, absolutely. And that's where these kinds of lab experiments become really valuable. By watching how different types of organisms decay today under controlled conditions, researchers can start to tease apart what they call ecological absences versus preservational absences.
Okay, what's the difference?
The ecological absence means the animal genuinely wasn't living in that ecosystem, just wasn't there. Preservational absence means it was there, maybe even common, but it just didn't fossilize well. Its body type or chemistry worked against it.
I got it.
So if you see in the lab that a certain type of small, low-protein organism consistently breaks down super fast, doesn't create those low oxygen zones, it suggests its absence in the fossil record might be more about poor preservation potential, not that it never existed in those ancient environments.
Right. So maybe lots of small, squishy things were wriggling around playing big roles in the ecosystem, but they just didn't have the biological oomph to leave a fossil trace.
That's the hypothesis this study supports, yes. They couldn't kickstart that crucial low oxygen preservation process.
That's a really powerful idea.
It is. But, the researchers are rightly cautious. They emphasize it's still a hypothesis. We can't, unfortunately, observe these ancient ecosystems directly to confirm it 100%.
Right, no time machines.
Yeah.
Good. Okay, but I know what some people listening might be thinking now. What about all the external stuff? The climate back then, the type of mud or sand they got buried in. Salty water versus fresh. Surely that matters too.
Oh, absolutely. Those external environmental factors are massively important, no question. Different sediments, muds, sands, clays, they definitely preserve things differently. And conditions like high salinity, really salty water, or very low temperatures, we know those can act like preservatives too. They slow down decay significantly.
So getting buried in a really salty lagoon or a freezing environment could also boost your fossilization chances?
Definitely. It adds another layer of complexity. But it's worth remembering, this specific study, their focus was really zooming in on the organism itself. How did its own body mass and chemistry influence that immediate chemical environment, particularly the oxygen levels, right after burial?
Okay.
They'd be the first to say this is just one piece of a much larger puzzle. There's still tons of research needed to understand how all these factors, internal biology, external environment, interact.
That makes total sense. You got to isolate variables sometimes to understand them. Okay, and one last thing on the experiments. Why those animals? Shrimp, snails, starfish, flatworms. Seems like an odd mix.
It's a good question. And Nora Cortese explained their thinking there. They chose those modern animals because they're seen as pretty good stand-ins or proxies for certain types of extinct creatures.
Stand-ins. How so?
Well, both in terms of their evolutionary relationships, their spot on the tree of life, and also their basic body composition. For example, the cuticle, the sort of external skeleton of a modern shrimp, share some important similarities with ancient arthropods.
Ah, so they're picking the closest living models they can find to try and understand what might have happened to things that lived hundreds of millions of years ago. Clever.
It's a way to make informed guesses, yeah.
Yeah.
Based on observable processes today.
Okay, so let's try and wrap this up. If we boil down this whole deep dive, what's the main takeaway message from this Lausanne study?
The core message is really that an animal's own body, its size and its protein content, is a surprisingly big factor in whether it gets fossilized. Bigger, protein-richer bodies seem to trigger these low oxygen conditions around themselves when they decay. And those conditions dramatically slow down decomposition, giving mineralization and other preservation processes a much better chance.
So the fossil record isn't just a neutral history book?
Not entirely, no. This suggests it's potentially biased towards organisms that were biologically predisposed to preservation because of their size and chemistry. It's less a perfect census, maybe more a reflection of who was built to last in a geological sense.
And that helps explain some patterns we see, like all those arthropods in the early periods. It wasn't maybe just that they were everywhere, but also that they were more likely to fossilize than, say, smaller, softer things living alongside them.
Precisely. It adds a crucial layer to how we interpret the fossils we find. We need to consider these preservational biases.
Absolutely fascinating. So it leaves you thinking, doesn't it? Given all this.
Yeah.
What other kinds of life might have existed back then? Maybe things that were small or soft or just didn't have much protein? Things that left almost no trace because their bodies just weren't suited for fossilization under normal conditions?
Yeah.
And how could future research even begin to find evidence for them?
maybe looking for subtle chemical clues in the rocks or studying unusual preservation environments. It boggles the mind thinking about the potential lost biodiversity, the chapters of life's history we might be missing.